will be the same. So let's say if I have this semi-permeable membrane, so when the net concentration is equalized, okay, which means to say that if I have if I've got 3 particles of water on each side, okay, the particles will still move, but it will be equal. So for example, one will move across here, the other one will move across here. Okay, it is not to say that there is no movement. There is still movement, just that the total movement is the same for the molecules. So when we talk about water potential, we have to compare in terms of the substances that are inside. So at this point in time, I want you to differentiate between the terminologies solvent and solute. Okay, so solvent is the substance that you dissolve things in. Okay, so this is the medium for dissolving. So think of some everyday examples that you have for such mediums. So example, I have got water. Okay, I also have in the morning when you drink, you have got your milk themselves. Okay, so the solute will be the substances that are used to dissolve. Okay. So for example, I will have things like my Milo that is going to dissolve in the milk or water. I will have my coffee powder, etc. Okay, so if let's say my solute concentration is high, which means I have got a lot of Milo powder. Okay, so let me repeat. If let's say my solute concentration is high, so I have got a lot of Milo in my water. It means to say that I have got a high concentration of solute. Okay, so which means over here it will be a hypertonic solution, okay, in which you have got large amount of solute particles. Okay, so since I have got a lot of solute particles, it means that my water concentration will be lower. Okay, conversely, if I only pour 1 teaspoon instead of the entire 1 tablespoon, let's say I only pour 1 teaspoon of the coffee into the water, it is extremely diluted. You actually taste almost like water. So that means you have got a very small amount of solute particles. So more water, therefore it is called hypotonic solution. Okay. Now, moving on to the next part here. So osmosis, when it takes place under these situations, how do you describe the processes themselves? Okay, so when I have got the same concentration in both X and Y, when I have got the same percentage, 10% sugar here, 10% sugar here, so which means that my water concentration will be 90% on both sides. So there will be no net movement. So which means to say that the movement of the water molecules will be the same to the left and to the right, and therefore I have got no net movement. Okay, so over here, if I have got a lower percentage of solute, so for example on my left side, okay, I have got 5% of sugar solution. So how much water solution do I have? If I just go by the total of 100%, I will have 95% that is water. Okay. On the other hand, over here at solution Y, I have got 90% that is water. So keep in mind, osmosis is a movement of water molecules from a region of higher water potential to lower water potential. So therefore, water will move from left to right. Okay, same thing for dilute solution. Dilute means very little. Solute means the substance that you're pouring to dissolve in. So for example, the tea powder or the coffee powder. Okay, so if I've got very little coffee powder, my water concentration will be high here. Okay, whereas in Y, my water concentration will be low. Okay, pure or distilled water indicates it is 100% water. So 100% water versus a less than 100% water in Y, my water movement will be from the left to the right side in the beaker itself. Okay, and thereafter there will be no net movement. Okay. Now, so these are the ones whereby we talk about isotonic, hypertonic, and hypotonic that we talked about earlier on. So when you have got isotonic solution, it means that the inside and outside are the same. They have got the same water potential. Okay, so you have got things that are called like your isotonic drinks, whereby it is supposed to match the electrolytes that you have in your body. So let's say after you go for a run, what happens? You end up sweating a lot. So you are losing electrolytes. Therefore, the purpose of the isotonic solution is to replace back the electrolytes that you have lost. Okay. In hypertonic solution, hyper is more. So which means that I have got more solute in the solution. Okay, so when I have got more solute in the solution, okay, so concentrated solution here, more solute, so which means my water potential is higher in the animal cell. So because water potential is higher in the animal cell, the water will move out via osmosis to the surroundings. So after some time, the cell is going to shrink, and that is called crenation or crenated. Okay, so conversely, in plants themselves, what happens if I have it in a concentrated solution? Okay, now plants is unique because they have a cell wall, and the cell wall, if you recall the topic on cells, it is rigid. Okay, so therefore, even when the water leaves the plant cell itself, the cell membrane is the one that is shrinking. Okay, so that is to say that my cell has become plasmolysed. It does not have the same spiky appearance that it do in animal cells. Okay, so a typical question for the plant cell part is, let's say for example, I have got a plant cell here. This is my cell wall, pardon my drawing, and the inner part I have got my cell membrane. Okay, so my cell membrane has shrunk, and the surrounding solution that I have is a starch solution. Okay, so after some time the shrinking, the typical question will be what is found in all these shaded areas where the plasma membrane has shrunk. Okay, so keeping in mind that my cell wall is fully permeable. Okay, so the outside here is a starch solution. Okay, so since it is fully permeable, my starch solution is able to enter into the cell wall and be at the area where it is green. Okay, so therefore when these areas are tested using iodine solution, they will test positive in a blue-black color. Okay, but the moment it hits the cell membrane area, okay, the plasma membrane, that is semi-permeable. So therefore the starch solution is unable to go through this area, and it will just be where it is at the green regions outside here. Okay, so let me just type it out. Alright, now let's go back to this part here. Okay, so at this part here, I have got a hypotonic solution. Okay, so for hypotonic solutions themselves here, it means to say that you have got a lower solute concentration. So this hypotonic solution, the dissolved solute is much lesser. So because it is much lesser, I will have a high water potential. Okay, so therefore the water is going to enter into the cells, and that will cause lysis in animal cells. Okay, you are not supposed to say that the cell has burst because it is not an appropriate term. Okay, now in plant cells, the same thing. I have got a cell wall. Okay, so because the cell wall, it is extremely strong, what happens is that the plant cell only becomes turgid. Okay, so when you draw out, you're supposed to draw the cell membrane to be very closely packed to the cell wall area, and the total change in the shape from the original to the expansion should not be drastic. Okay, it is only the shape of here, this region, that should be closer to each other. Okay, so why do all these things take place is because we have got what we call surface area to volume ratio. Okay, so an everyday example will be, for example, if you are going to cook a dish, you do not just throw the entire potato into the pot. So what do? You cut it up into small pieces. So the smaller the pieces, the faster the rate of cooking. Okay? So why is that so? Is because the smaller the piece, the surface area to volume ratio is actually larger. Okay? So look at this small piece here. The ratio will be 6:1. Compared to a large one, it is a ratio of 2:1. Okay, so therefore the smaller the size, you will have greater surface area, so therefore you have got faster movement of substances. Okay, so why is faster movement of substances important is this red box you have here. It enables nutrients and oxygen to diffuse quickly into the cell, okay, which is of importance because the cell requires it for respiration, photosynthesis, and other modes of energy making areas. At the same time, the cell also produces waste, not just in plant cells or animal cells, but in almost any type of cells. And the waste production to go out of the cells will be quicker, and that is required to prevent toxicity from building up. Okay, so what are some of the adaptations that is made possible for these? Okay, we covered some of these under the topic for cells themselves. So root hair cell, you have got an elongated protrusion for increased surface area to volume ratio. Red blood cells, you have got biconcave shape. Okay, so to recap, your biconcave shape actually looks like that. Okay, so both sides they are concave, so that will increase the surface area to volume ratio for these cells.